So, good afternoon, everybody. My name is Boelo Schuur. I'm the Scientific Vice President of the European Federation of Chemical Engineering, the EFCE. I've been the board of the EFCE since January last year, and I'm a chemical engineer by training. I received my degrees from the University of Groningen in the Netherlands, and I built my career in academia. And since 2011, I've been working at the University of Twente, where currently I'm a full professor, and my chair is called Sustainable Separation Technology. I'm very happy to be involved in the introduction of this webinar on chemical engineering applied to medicine. Specifically, chemical engineering contributions to new materials, technologies, and modeling methods with applications in the diagnostics, pharmaceuticals, and artificial organs. I'm looking forward to participate in this. My role is to introduce the Spotlight Talks and the EFCE, which I will do now. I will start with the Spotlight Talks. Spotlight Talk series is now the 6th year that we organized a series of Spotlight Talk sessions. And after the COVID pandemic, we have decided, based on the success of these sessions, to maintain the Spotlight Talks on the calendar, even though we have now our regular in-person meetings also taking place again. This year, from 8th till the 10th of September, we have the European Conference on Chemical Engineering, the ECCE ECAP in Lisbon. And for that reason, we don't have a fall series, and we only have the spring series. But in this spring, we will have 10 Spotlight Talk sessions organized by 11 of our working parties and sections, including chemical engineering applied to medicine, sustainability, chemical reaction engineering, process intensification, comminution and classification, mechanics of particulate solids, fluid separation, polymer reaction engineering, static electricity, crystallization, and the early career chemical engineers. From today, the 14th of May, through the 22nd of May, we may enjoy these short sessions of 3 or 4 talks focused on specific topics by leading industrial and academic experts. The complete program is available on the EFCE website, efce.info, and you are encouraged to explore the other sessions as well in the coming week. The EFCE promotes scientific collaboration and supports the work of chemical engineers in 30 European countries, representing more than 100,000 of them in the whole of Europe. EFCE's working parties and sections cover all major specific aspects of chemical engineering and are the core of the organization, forming the scientific engine that drives many of the EFCE's activities. They provide an important forum for technical exchange and networking among chemical engineers in Europe. In each of our working parties and sections, highly skilled engineers gather. And I would like to invite everybody who is not a member of a section or a working party to have a look at the website and to explore our scientific groups. Maybe you are interested in joining one of our sections, who are open for everybody to join. Joining a working party is on invitation only, but if you see that your country is underrepresented and you feel you are one of the potential representatives, or you know a highly skilled individual in that domain of chemical engineering, then please reach out to us and to your national member association. And then we can start seeing opportunities together. Before concluding, I would like to thank all the people who worked hard in the working party sections and in the EFCE in general for this initiative to happen again and to have a next successful series. In particular, Martina Poe from Toulouse, who is doing most of the conceptual and setting up work and a lot of practical activities. I thank you for your attention, and I wish all the speakers and the attendees a fruitful and successful webinar. And I will now give the word to Tomasz Sosnowski, the chair of the section Chemical Engineering and Applied to Medicine, to introduce the first speaker of this webinar. Thank you.
Thank you very much, Boelo. Yes, I want to also welcome everybody who is attending this webinar. And as you know, it's organized by our section, which is called Chemical Engineering as Applied to Medicine. I have a few words about this section. We have 5 minutes just for the introduction. As you know, my name is Tomasz Sosnowski. I work at Warsaw University of Technology in the Faculty of Chemical and Process Engineering. This is my background. I'm in chemical engineering, my original background, but from many years I'm just interested in applying these rules of chemical engineering, these methods, to improve drug delivery, especially drug delivery to the lungs. And this very well fits to the topic of the section. And the faculty, I'm also a vice dean for the scientific research and development at the moment. So, okay, just I want to know a little bit about me. And right now, I will trying to share the screen just to show a little bit about chemical engineering. Maybe starting from the beginning. This section was based on the, knowing that the actual human body is a big bioreactor, very complicated one, but still it can be understood as a special process system when we have different organs connected one with another by blood flow as a pipeline of the system, and other functions like pumping, like mass exchange through the membranes, filtration, and so on. Of course, this is a simplification, but this is a main idea which actually made us to think about chemical engineering as a good tool for understanding what's going on in our body, and actually how can we help medicine to be to progress. And so actually at the beginning is the idea of using the toolkits of chemical engineering to solve selected problems of physiology and medicine. And actually these problems can be divided to different fields. For example, systems engineering and modeling in physiology, also including machine learning and artificial intelligence, but also other methods like CFD. Okay, and we can apply them to metabolism, pharmacokinetics, and pharmacodynamics, diagnostics, also to assessment of toxicology. Also, transport phenomena in the human body. This is a very important thing just to understand how drugs can be delivered to the system, and how can we improve the therapy. Also, a lot of things is to do about new nano and micro materials, dispersed systems, but not only, also surface science, which help us to develop new devices, to design some artificial organs, and also drug carriers. So altogether, we know that chemical engineering is the discipline that works on multi-scale approach. So we are also working on multi-scale approach from the molecular side, let's say, scale up to big, also pharmaceutical factories. But actually, we are not in the pharmaceutical industry with this topic. We are more in the development of the tools that are working directly with the human body. So the timeline of the section is like this. We are established more than 2 years ago in December 22 in Paris. We have a kickoff meeting during the 4th European Forum on New Technologies, which was devoted to chemical engineering as applied to medicine for the first time within the EFCE. Then 2 years ago, we had the first spotlight talks, which was entitled very general, just chemical engineering as applied to medicine's current challenges and opportunities. We have 4 talks at that time. Then we have dedicated session during the 2023 European Congress of Chemical Engineering, together with European Congress with Applied Biotechnology, which was held in Berlin at that time. And then we organized the first conference of our section, which was in May 2024, which was in Salerno in Italy. And we gathered almost 50 people there. We have also a book of abstracts from this event. It was a very successful event, and we are growing our community. And then this year, autumn 24 and spring 25, we published set of thematic papers in Physical Sciences Reviews, which is the journal edited by the Greuter. And we are preparing the book, which is, you see the title of the screen, and it will be published by this same editor probably this year or in the beginning of the next year. And finishing that, we are today on May 15, 2025, with the second Spotlight Talks devoted to this topic. So now I want to introduce the first speaker of our talk. Excuse me, just a second. The first speaker of our talk will be Lucia Baldino from University of Salerno, and she will talk us about the supercritical fluids and nanosomes, applications and perspectives in precision nanomedicine. I just want to add that if you have any questions, please put them in Q&A, questions and answers window, because we will answer only, we'll talk about only in this way. We are not allowed to talk because we have many people on the webinar, so just please your questions to the Q&A. Okay, thank you. And I want to ask Lucia for presenting her talk.
Thank you. Just a moment, please. Okay. Do you see my screen?
Yes, we do.
Well, it's okay. Okay, so thank you. Good afternoon, everybody. Thanks, Tomasz, for your kind introduction. And thanks to all the organizers of this Spotlight Talk related to the chemical engineering as applied to medicine section. My talk of today deals with the application of a new continuously green supercritical CO2-assisted process for the production of nanosomes that are particularly interesting nanocarriers for precision medicine application. As we can read from a recent communication published on Nature Chemical Engineering, a bright future in medicine for chemical engineers is expected, thanks to the background, to the approach of chemical engineering that can be useful for the design and the realization of new medical devices, treatments, and strategies also in medicine, of course, in collaboration with biologists, medical doctors, and so on. But the first question is, what means nanomedicine and why is small different? So, nanomedicine is the application of nanotechnology to achieve innovation in healthcare. And the important thing is that we need materials at nanoscale that are characterized by different properties with respect to the same materials at a larger scale. But it's important to work at this scale because most of the biological phenomena and mechanisms occur at this scale. So in order to interact, we need the same scale. Nanomedicine impacts all the field of medicine, from diagnostics to medical imaging, vaccines, regenerative medicine, and nanotherapeutics. So, looking at this last field, in the last years, many types of lipid-based nanocarriers for drug delivery have been proposed both in the literature and on the market. Among them, liposomes are frequently proposed and studied. Why? Because liposomes show a cell-like structure constituted of an external double lipid layer and an internal aqueous core. So, thanks to this organization, they can encapsulate both hydrophilic and lipophilic drugs, and they show an excellent biocompatibility with our body. However, liposomes suffer from some limits, in particular due to their starting materials, phospholipids. They can degrade due to oxidative phenomena, and therefore they require storage at low temperature. Moreover, since they are typically ionic, they can be partly toxic for our body, and both raw materials and processes involved in their preparation are expensive. So, in order to try to overcome these limits, neosomes have been proposed in the literature and also the market. As we can see from this picture, the structural organization of neosomes is very similar to the one of liposomes, but the main difference is represented by the starting materials that in this case are non-ionic compounds like surfactants. For this reason, neosomes show a longer physicochemical stability even at room temperature. They show a reduced toxicity, lower cost, and raw materials are readily available. Currently, neosomes have been tested to encapsulate several bioactive compounds, proteins, peptides, vaccine, natural products, and they have been also tested via different routes of administration, intravenous, dermal, ocular, and so on. However, both liposomes and neosomes show some limitations when used as topic or transdermal drug carriers due to their rigid nature. And therefore, drug cannot pass efficiently in the deeper layer of our skin. Therefore, in order to overcome this problem, transversomes have been proposed. They are defined as ultra-deformable vesicles thanks to the addition of edge activators to the phospholipid bilayer. Generally, edge activators are represented by surfactants. So, by adding surfactants in the lipidic layer of transversomes, we can create a sort of discontinuity that favors the deformability of this kind of vesicle, and therefore they can pass through the so-called stratum corneum of our skin through this schematic general mechanism. So they can deform it passing through stratum corneum, and the driving force is represented by the humidity gradient between the external and internal environment of our skin. However, the current and conventional nanosome production methods, such as thin film addition method, microfluidics, freeze drying, and so on, suffer from several limits. These are all batch and time-consuming processes. They use organic solvents, and therefore post-processing steps of purification are required to obtain safe vesicles for pharmaceutical application. Moreover, they can assure a reduced control of size and shape of the final vesicle. In this case, several post-processing steps of extrusion and filtration are required to obtain the proper particle size distribution, and generally they lead to low drug encapsulation efficiency. So some years ago in our research group, we thought about in which way we can improve the production methods of nanosomes by applying supercritical fluids, and thus supersome process was born. But the first question is, what is a supercritical fluid? A supercritical fluid is a substance above its critical pressure and temperature. And in this condition, a supercritical fluid shows hybrid properties between a liquid and a gas. In particular, a density closer to the one of a liquid and a diffusivity and viscosity closer to the ones of a gas. Among the potential supercritical fluids, carbon dioxide is the most frequently used because it is characterized by a mild critical temperature and pressure. Moreover, it is cheap and inert. An opposite example can be water, and as you can see from the table, it shows a more than double value of critical temperature and a relatively high critical pressure. Therefore, processes assisted by supercritical water are expensive. The main relevant properties of supercritical carbon dioxide are variable solvent power through its density by changing the operating temperature and pressure, solventless products because by a simple depressurization, we can remove CO2 from the system, gas-like mass transfer, and therefore the processes assisted by supercritical carbon dioxide are faster with respect to the corresponding traditional ones. Negligible surface tension. This property is important when we want to produce nanostructured material. We can avoid the collapse of this structure at nanoscale, and large chemical affinity with organic solvents. Therefore, CO2 is a good anti-solvent. Here I reported a schematic representation of Supersom's lab-scale plant layout. In particular, if you look on the right, the core of the plant is the high-pressure vessel, this one, in which we have the atomization of water or of a water solution, I mean water plus the active pharmaceutical ingredient. In the same vessel, we have the feeding of a gas-expanded liquid formed by the ethanolic solution of phospholipids or surfactants, depending on the kind of nanosome we want to produce, plus supercritical carbon dioxide. These two lines go to this saturator or static mixer, that is a vessel in which we have packing material in order to favor the interaction and the interposition of the small molecules of CO2 between the molecules of the liquid in order to reduce drastically the cohesive force of the liquid in order to obtain a gas expanded liquid. So in the high pressure vessel, we have gas-like mass transfer phenomena, and therefore on the fly, water droplets are surrounded by a first layer of lipids thanks to the interaction of the polar heads of lipids with water, forming the so-called reverse micelles. When these micelles fall at the bottom of the vessel, where an excess of water and lipids is present, we have the formation of the second lipidic layer forming the vesicles, and in continuous way we can withdraw from the bottom of the vessel the aqueous suspension of vesicles. Here I reported a sort of detail of what happens inside a pressure vessel. In the table, the operating parameters that we generally use for nanosome production, these parameters, pressure, temperature, and liquid flow rates, have been optimized in several previous works of the research group. So, summarizing, main advantages of Supersome process are one-step continuous process, very fast process, control of vesicle mean size by changing thermodynamics and kinetics of the process, solventless vesicles, high drug encapsulation efficiency. So now we can move to some examples of nanosomes produced by Supersome process, of course, from a chemical engineering point of view. The first example is the production of stealth liposomes. The first question is why we should produce stealth liposomes. We know that liposomes have the cellular-like structure, so they show an excellent biocompatibility with the human body. However, when used as drug carrier, they can be recognized as non-self by our organism and therefore eliminated. So in order to avoid this problem, generally the external surface of liposomes is coated by using hydrophilic macromolecules or biopolymers like polyethylene glycol in order to create a thin water layer around the liposomes, and operating in this manner, opsonists, that are proteins of our plasma, cannot recognize liposomes, and therefore they have a longer residence time to reach the target site and release the drug. So we produced liposomes by Supersomes process. We prepared two different solutions. The first one constituted of water plus vancomycin, that is an antibiotic, in this case used as a model bioactive compound, and ethanol plus phosphatidylcholine. After processing, we obtained liposomes, and then we performed the so-called pegylation by using the post-insertion method. We use this kind of PEG, that is the most frequently proposed in the literature for this application, and we optimize this step in terms of concentration of PEG and temperature, duration, and mixing speed of the pegylation step. And here you can see the optimized parameters obtained from this step. And stealth liposomes were characterized in terms of morphology. As you can see from this scanning electron microscopy image, we obtain liposomes characterized by a spherical, regular, nanometric mean dimension, around 200 nanometers. We achieved a vancomycin encapsulation efficiency up to 89%. And also we checked the vancomycin release time and profile from stealth liposomes. So obtained that in the case of stealth liposomes, we detect an increase in the release time up to 20 hours, with an increase of 10 hours with respect to the release from simple liposomes. So in this case, we also verify the mass transfer resistance opposed by the external coating of. The other example is related to the co-encapsulation of curcumin and vitamin D3 in liposomes. In this case, we used a mixing between two types of phospholipids, or phosphatidylcholine, hydrogenated and non-hydrogenated phosphatidylcholine obtained from soy and egg yolk. And we selected as bioactive compounds curcumin and vitamin D3 because both are characterized by nutraceutical properties and also anti-tumoral and other pharmaceutical properties. And their nanoencapsulation can improve their bioavailability and biodistribution in our body. And as you can see from this graphical abstract, in both cases, we obtained nanometric liposomes by scanning electron microscopy analysis. But when we load both bioactive compounds, we detected a slight reduction of curcumin encapsulation efficiency from 95 to 84% due to the presence of vitamin D3, since both are lipophilic compounds. So there was a sort of competition in the same phospholipidic layer. However, in both cases, curcumin release profiles were comparable and diffusion governed the release mechanism. In this other example, we produced ketosomes, starting from neosomes. So, first of all, we produced neosomes by supersomes using for starting formulation span-18 and twin-18, these two surfactants, in a weight ratio equal to 19 to 10 weight percent. This formulation was optimized in a previous. Then we performed by using a post-processing method, the coating of neosomes by using medium molecular weight chitosan. Thanks to the electrostatic interaction between negatively charged neosomes and positively charged chitosan. And we checked the proper coating of neosomes with chitosan looking at the zeta potential value. Indeed, it changes from a negative value from a positive value. So we characterized the chitosomes in terms of morphology and other properties. In particular, they showed a regular spherical nanometrical morphology with a mean size around 200 nanometers. We also encapsulated in it doxycycline iclate, that is an antibiotic, reaching an encapsulation efficiency up to 83%. And then we test the performance of ketosome, in order to validate the pH responsive drug release. So we prepared 3 different liquid medium at different pH in order to simulate intestine small colonic colonic, in green colonic conditions. That was our goal, and we verified that most of the doxycycline release was obtained in the last region, a pH 6.8. That was our purpose. So thanks to this characterization, ketosomes could represent promising candidates for colonic disease treatments. In this last example, we produce transversons for anti-cancer treatment, and we load this kind of vesicles with a peptide synthesized ad hoc by our colleagues of the National Cancer Institute Pascale Foundation of Naples. And as you can see, after processing, we obtain nanometric transversons with a unimodal size distribution, a mean size, a mean diameter around 150 nanometers, a zeta potential value minus 19 millivolt, suggesting stable vesicles. Moreover, we also reached a peptide encapsulation efficiency up to 92%. By using nanocytes, and so by performing nanotracking analysis, we demonstrated that most of vesicles were in the dimensional range between 0 and 200 nanometers, in line with SEM and DLS analysis, and particle concentration reached 10 to the 11th particles per ml. Here we can see from this dynamic frame the regular shape of the produced transversons. And currently, biological tests with cells are in progress. So, concluding, up to now, we successfully produced by Supersons different types of nanosomes, liposomes, neosomes, transversons, loaded with different bioactive compounds of different nature, hydrophilic, hydrophobic, and we also performed the coating of vesicles by using different biopolymers depending on the specific application. Currently, we are working at the loading of mRNA, and the next steps will be the loading of other types of nanoparticles, also inorganic nanoparticles, and different active pharmaceutical ingredients, and we also try the coating with more complex biomolecules like proteins, antibodies, and so on. In perspective, in the next future, we will perform the encapsulation of nanosomes in hydrogels in order to protect them when administered and to obtain a final targeted delivery. Of course, in order to perform all these experimental steps, we are open for collaboration. So I would like to thank all the research group, my PhD student Sonia Sarnelli, and all the master's degree students that help us in performing the experimental part. And of course, I would like to thank all the audience for the kind attention. Thank you.
Thank you very much, Professor Baldino. Thank you for this interesting talk. We don't have questions in Q&A, but if I may, I just want to ask you just a single question. You showed this CO2-assisted generation of neosomes or other somes, let's say, and how well you can control the size. I mean, of course, you can control because you show, but this is very narrow or is multidisperse. How you can answer to that? It's about the polydispersity of the particles or no particles. This neosomes or transferosomes you produce in your technology.
Thank you for this very interesting question. Generally, after processing, we obtain directly unimodal size distribution vesicles with a PDI around 0.2, that is the target for pharmaceutical application, without performing other post-processing steps of extrusion or sonication. And we can obtain this regular size distribution by controlling the thermodynamics of the process in terms of pressure and temperature, and therefore supercritical carbon dioxide properties, but also by controlling the relative flow rates between the water solution, the ethanolic solution of lipids, and also the injector nozzle diameter.
Yeah.
So by changing these parameters, we can modulate the final size of our vesicles depending on the kind of application, but also depending on the kind and molecular weight of the drug we want to load. Of course,
Yes.
bigger molecules require larger vesicles.
Of course. Sure. We have one question from the audience. I just read it. Thank you for the presentation. Very interesting. In the supercritical CO2 process, CO2 can be recycled, or if this is the first question, if it can be recycled, and then if yes, how to isolate ethanol from the ethanol plus CO2?
Okay. In our case, we have a lab scale plant, so generally we release CO2 in atmosphere. But this is not a problem because the CO2 we use comes from a natural source. So the net balance of CO2 in the environment is the same. But of course, if we think an industrial process, we can recycle CO2 in a closed loop by using pumps, heat exchangers, and so on. About the ethanol
separation
recovery,
between
yes, okay. CO2 and ethanol can be separated in a downstream separator after depressurization of the system. Operating this way, we obtain gaseous CO2 from the top of the separator and liquid ethanol from the bottom, so we can recover ethanol and recycling it for other application.
Yes, I think this is a very, very good answer. Thank you, Lucia. Thank you again. We have to move on. So now I want to ask Professor Maria Norberta de Pinho from University of Lisbon, Portugal, to present her talk about mixed matrix membranes. The title is longer, but Maria will say that everything to us. Yes. Yes, thank you. We see the.
Good afternoon. Thank you very much for the invitation, and thank you very much for this to put up all these beautiful spotlight talks. Thank you very much to Tomasz, to Martin, and thank you. And I will be talking about mixed matrix membranes. And these mixed matrix membranes, it's to be used in hemodialysis. And hemodialysis is a medical practice where we want to remove the uremic toxins in the patients that don't have the kidneys functioning well. So the membranes in these hemodialyzers, they are usually ultrafiltration membranes, and these membranes have to remove the uremic toxins, but have to retain the vital components of the blood, mainly the plasma proteins. So these membranes have to have an adequate molecular weight cutoff, so the toxins can permeate and the other vital components have to be retained. Okay, and talking about toxins, we have three groups of uremic toxins, and we have the low molecular weight water-soluble uremic toxins that are the toxins, in fact, that are removed in the current state of hemodialysis. They are removed. But there is a third group that it's on the bottom of this table that are the protein-bound uremic toxins. And these protein-bound uremic toxins, they are not removed in the current hemodialysis. And they are a problem because they accumulate in the blood and they bring a lot of serious medical problems. So we will be concentrating in one of these uremic toxins, the picrisyl sulfate, because it binds to albumin, and then it stays in the system, in the blood, and does not get removed. So our objective, it will be to address the way to remove this picrisyl sulfate from the human serum albumin. And the picrisyl sulfate, it's usually bound by van der Waals forces and electrostatic interactions and hydrogen bonding. Yes. So, as I said, the picrisyl, it's a small molecule, but when it's bound to the albumin or to the other plasma proteins, it becomes a large molecule and it's retained by the ultrafiltration membranes. Okay, so our objective, it will be to have competitors. And our competitors, it will be the metal organic frameworks, the MOFs, that it will be incorporated in membranes, and it will be competing with the albumin for the absorption of picrisyl in this case, or to other uremic toxins. Okay, so, and why are we speaking with the membranes with such a long name, cellulose acetate, silica, and metal organic frameworks? Yes, till the 1970s, all the membranes used in hemodialysis were regenerated cellulose or cellulosic materials. And there were some hemocompatibility problems, particularly with the complement protein interaction. And so other polymers start being used, and like the polysulfones, that they are very used nowadays. And but the cellulose acetate, that it's derived from the cellulose materials, but does not have this problem of hemocompatibility because of the substitution of the OH groups by acetyl groups. And besides the cellulose acetate, it's a very well-known polymer to be used with very good film-forming properties and with a lot of flexibility as membranes goes. Then the cellulose acetate have also strong limitations, and the chemical resistance, it's one of these limitations. So for that, we did the membranes through the phase inversion technique coupled with sol gel for the introduction of silica. And the sol gel method that it's coupled with the phase inversion technique is done in acidic conditions in order to have the silica covalently bound to the cellulose acetate. So in these acidic conditions, we are sure that we have the silica covalently bound to the cellulose acetate matrix, because we have, by the ATR-TIR, we have the evidence that we have this peak, the COC peak, that it's clear in all the membranes that have synthesized, that it's clear that the silica is bound to the cellulose acetate. So with this in mind, we introduce now the MOF, and the MOFs to be special sorbents with preferential adsorption properties with regard to the proteins and the albumin. Okay, so here we have the MOF that we synthesized. It's a very common MOF, WO66, and this MOF was introduced in the casting solutions. But before, why this MOF? And it's exactly this MOF is a competitor to the bound to the uremic toxins to the BSA. Yes, here we have the evidence in this study of adsorption of becquerel in the presence of the MOF and the bovine serum albumin. I should say that we are using as a model the bovine serum albumin because it's very similar to the human serum albumin in terms of the adsorption effects. Okay, so when we add the MOF, here we see that 5 milligrams per milliliter is enough to immediately decrease the becquerel that it's bound to the protein from 80% to 40%. So that it was the reason to choose this MOF, and we went ahead and making membranes and making the synthesis of membranes with this MOF. Okay, so as I said, the MOF is dispersed in the casting solutions, and the casting solutions are cellulose, acetate, silica, that because we had to the casting solution that are made of the polymer, cellulose acetate, and the system of solvents that is the formamide and acetone, and we disperse the MOF, and we add the precursor of the silica, the TEOS, that it's added to the casting solution, and having these, as I said, in acidic conditions, we have the precursor bound to the or the silica bound, covalently bound to the cellulose acetate. Okay, so here we have casting solutions for membrane without MOF and membranes with 1%, 1.5%, and 2% and 2.5% of MOF. So the casting conditions were evaporation times of 30 seconds and water coagulation bath. It is water at 0 degrees. Okay, so then here we have the characterization of the membranes, and all these membranes, they have hydraulic permeabilities that generally there is a general trend of increasing with the MOF percentage, and particularly after 1.5%, 2%, and 2.5%. The molecular weight cutoff of the membranes, obviously, they increase with the increase of the MOF content. Okay, and these membranes, practically they let all the salts go through, very small rejections, not only for NaCl, as for the sodium sulfate, and these really it's typical of ultrafiltration membranes. And they have for the pecrezyl, free pecrezyl, they have low rejection coefficients. Okay, and then as we see here, the fluxes, the water fluxes that they are in blue, and they are particularly higher for the membranes with 1.5% of MOF, and they are higher for the other two, but they decrease somehow after the 1.5% of addition of MOF. Okay, and for the other solutes, as we could see, there is also a change when we have the MOF, and we have these rejection coefficients increasing slightly. Okay. And now, about the adsorption isotherms of pecrezyl and pecrezyl sulfate, as we can see here, we have the adsorption isotherms for the MOF in the free form, and then for the membranes, for the four membranes with the different contents of MOF. And we have a completely different adsorption pattern. We have a isotherm, a Freundlich isotherm for the free MOF, and then we have Langmuir isotherms for the membranes with the MOF. And as we can see here, the limit of the adsorption of pecrezyl, it's situated below 5 milligrams per gram of pecrezyl, of MOF. Sorry about that. Okay, and now for the membranes, the adsorption characteristics for the membranes with different concentrations of MOF, and as we can see here, the membranes with higher content of MOF, they have adsorption, different characteristics of adsorption of pecrezyl, but I think these the 2 and 2.5% are some difficulties here in the dispersion of the MOF. So we go ahead and see it in a different form. We have here the adsorption adsorption isotherms. Now, in the presence of the free MOF, and for the membranes with different concentrations, but the pecrezyl together with the BSA, they are present. And as we can see here, that the adsorption of BSA, it's below the adsorption of the UEO 66, but the membranes with incorporation of MOF, they are in a range slightly, some of them have slightly upper range from the BSA, but there is not a very pronounced difference as for the free pecrezyl. Okay, so in conclusion, in fact, we have a MOF that brings down the pecrezyl adsorbed to the protein from 80% to to 40%. And the membranes showed that the addition of MOF and the increase of its content gives higher hydraulic permeabilities, ranging from 14 to 34 kilograms per hour per square meter per bar, and higher molecular weight cutoffs are brought by the inclusion of the MOF. Okay, and so there is a decrease in the rejection coefficient for membranes with higher MOF content, and we think that the membrane with 1.5 and 2% and 2.5% will be the best ones for the filtration or the competition with the albumin for the removal of the pecresil, as they combine lowest toxin rejection with high hydraulic permeabilities and high fluxes of the permeation. Okay, so I would like to see that our future work, it will be trying to get other MOFs and to see if the mixed matrix membranes with new adsorbents, it will have better removal of these uremic toxins that bound to the proteins. And also another thing that I didn't talk, but it will be investigated, it's the removal of middle-sized uremic toxins that are not a big problem like the PBUTs, but they are also in some cases a problem of staying in the bloodstream and not permeating through the hemodialysis membranes. Yes, and I would like to thank the Foundation for Science and Technology for the founding of the two centers that contribute to this work, and to the research team made by Diogo Barradas and Miguel Pereira da Silva and Moisés Pinto. Thank you.
Thank you very much, Maria. Very interesting talk. I will see if we have any questions from the audience. I cannot see anything right now. You work on the flat membranes, right?
Yes, exactly. Yes. Yeah, very good question, because the hemodialyzers, they are hollow fibers.
Yes, yes, but of course this is a basic study, so of course you start from the well-controlled conditions.
There is still a long way to, but we are involved in another project, a European project, but with the lung. And in this project with the other partners, we are doing hollow fibers.
Okay.
And, so probably in the future we will be addressing this.
Okay, thank you very much.
There are some hemodialyzers for children that they use flat sheet membranes. Yeah.
Okay, thank you very much. We have a little bit late, so I think that we will skip to another talk. Thank you again. And another talk will be given by Professor Davide Manca from Milan Polytechnic, Italy. And this talk and the next talk will be about modeling, actually, about machine learning. So we are very happy that we combine some experimental studies that we've seen before with some computational work that we will see in the two next presentation. Davide, please, the floor is yours.
Thank you, Tomasz, and thank you to all of the attendants to this seminar. And let us go with the agenda. I will present and I will deal about two different data sets. Let us say big data, because this is the trend of today, dealing with big data. And I will deal about two applied case studies. First of all, we can say that data science and machine learning are transforming healthcare by also using and, let's say, exploiting the power of information which is contained into these large data sets. And this is also intended for preventive healthcare. And pharmaceutical companies are very interested not only in synthesizing new drugs, but also nutraceuticals or other forms of vitamins, minerals, probiotics, and so on, because they are easier. They usually take a much lower time to product, and the regulation is significantly lower, and so it is much easier to go on the market. And so we can also see that people, at least for instance in the USA, let's say 4 or 5 years ago, they were spending in excess of 10-12 billion of dollars in herbal supplements. So this trend is continuously increasing year after year. So I will deal two case studies. The first one will be about diet and illnesses and the link for possible nutraceuticals that might help reducing the impact of these diseases, and the other will be about the machine learning method for LDL, cholesterol, low density cholesterol assessment. So first of all, let us focus on the first data set. It comes from NANES. It is available online without even registration. It's open. It is made available by the USA, and NANES means National Health and Nutrition Examination Survey. And it started in 1999, and every 2 years it is updated and it increases. It deals about every year they examine about 5,000 participants, which are invited on a basis of a 4-stage selection that goes from counties of the USA, going to segments, households, up to the single persons. And these persons are actually not patients, but individuals, normal people that are covered in terms of race, age, cultural background, and they try to depict and cover the distribution of individuals in the USA. Once they are invited, they are asked a lot of questions, and they have to comply with a number of questionnaires. And let us first focus on diet and illnesses. Every participant provides a detailed, let's say, list of nutrients intake, daily intake, and also supplements. And they are asked to answer a number of questions, and they are subject also to a blood exam in order to have an overall idea of what are their kind of life, quality of life, and health issues. So they answer to dietary nutrition intake, and they report about foods and beverages, but also possible supplements intake in terms of vitamins, minerals, herbals, and so on. By doing so, these bits of information are processed in order to understand the intake in terms of vitamins, in terms of minerals, all over as a sum of those two kinds of intakes. And they are also asked for a number of questions. I just reported here 3 different questions, They are about what happened in the last 30 days in terms of, for instance, any problem in terms of cold or intestinal stomach illness or pneumonia. And okay, let us see what they answer and possible interaction with their, let's say, habit in terms of health and intake, daily intake. So we focused on adult individuals, which means over 20 years old. We identified outliers and we applied, first of all, a number of statistical tools and other techniques in order to extract information. The objective was understand whether a different total nutrient intake is associated with a different health status. So just to have an indication that might make the difference between more healthy people respect to people who have problems with their health. So at the end, we were able to assess that there is a distinction between individuals that had problems with stomach and intestinal illness in the last 30 days, respect to those that didn't. The two distributions, the two groups are statistically different. How did we obtain this result? This is the anticipation of what we carried out. And this was achieved by means of a PCA, principal component analysis, that tries to identify the more important, the most important details in terms of information that a given amount of data carries with its content. And we were also able to see that according to the 15 most important vitamins, minerals, nutritional issues, the first PCA, the first component, was able to describe a large amount of information, because this is a monotonically decreasing trend, and the first one was able to describe the 55% of the data set variance. So we focused on the first component, and as you can see here on the right, there was a significant difference between the people who didn't suffer from any problem in terms of stomach and intestinal illness respect to those that suffered. There were about 12,000 people who didn't and 1,000 people who had problems. So are these groups statistically different? They were subjected to a Mann-Whitney U test, and they proved that the distributions were different. And so we could also extract the first important information, which is higher values of the principal component number 1 can be associated with a lower inflammation level. So let us try to understand what PC1 contains, which kind of detail of information contains inside. And PC1 as any principal component is a linear combination of different features weighted by different weights. And here you can see that they are ordered from the higher to the lower one. And you see that selenium, calcium, phosphorus, zinc, iron, and magnesium were playing the most important role that allows us to say approximately, or to make this hypothesis that these elements play a role in, let's say, preserving individuals from intestinal and stomach diseases. So we were also trying to see the interaction between pairs, for instance, phosphorus and magnesium, phosphorus and selenium, selenium and magnesium. And by looking at the intake of every individual, we saw that the recommended dietary allowances, that are daily allowances, were overcome and were above the average threshold in terms of selenium, phosphorus, and magnesium, with this excess for the people who didn't suffer from problems. So it means that people that were having an intake of selenium, phosphorus, and magnesium, which was higher than the RDA, were suffering less of stomach and intestinal disease respect to those that were below. So this is a good indication that suggests that featuring and preparing a nutraceutical based on these elements should allow an improvement, should provide an improvement in this health status. And in fact, also looking at the literature, we were able to see that selenium and magnesium are considered very important and to play a role in reducing the inflammation of the intestinal duct. And for this reason, selenium, phosphorus, and magnesium are, as I said before, a good candidates for obtaining a selected nutraceutical that is targeting the inflammation reduction of intestinal problems. The other topic that we focused based on machine learning was about the LDL assessment, a model that allows us to calculate the low density cholesterol, that is the type of cholesterol that is most closely associated with cardiovascular disease risk, and therefore it's the most important to monitor. Usually when we go and have a blood exam, total cholesterol, high density cholesterol, and triglycerides are measured experimentally. Vice versa, LDL is usually never measured, but is inferred from the previous three values. By knowing the experimental value of total cholesterol, HDL, and triglycerides, we can obtain LDL by means of some formulas. The most common are the Friedewald, that is a little old, let's say, a bit old, and also a more recent NIH2. They have some limits, however, that can be shown in a matter of few minutes. So, to understand their limits and to see if any new model can be achieved, we used a very recent data set that is available in the public domain and is an anonymous high triglyceride and LDL values data set. It covers almost 26,000 individuals that anonymized, and they report the age, gender, total cholesterol, HDL, triglycerides, non-HDL, direct LDL. It means measured in the laboratory. So these individuals were measured, the cholesterol, and also the NIH2, the most recent equation, is also reported. And after using this data set and subjecting it and undergoing an outlier removal, we were able to identify in excess of 24,000 individuals. So there wasn't a significant reduction. And here we can see, I anticipated why the Friedewald on the left and the NIH2 on the right are equations that allow us to predict LDL but they are affected by a consistent bias because both equations tend to underestimate LDL values. And this is not good at all because a high value of LDL is not a good point because would bring an increased risk in cardiovascular disease. And you can also see that the percentage of the points of single individuals who are above the equivalence line between the measured and the evaluated value for LDL is quite high, 94% in Friedewald and 88, 89% for the NIH2. So I would say that there is a significant limitation using those equations. So first of all, we tried to extract information, identify, let's say, the model by means of neural networks. First of all, we used rather simplified neural networks with just one output, the LDL value, the 5 inputs that were reported before, and only 2 hidden layers. And we were using different activation functions, even though the ReLU or ReLU function was the best. And also dropout rate from 0 to 30%. From this selection, we saw that we were able to have a significant reduction in the mean absolute error from 36 given by the best available conventional equation to 16. And then we also tried to go in, let us say, a kind of deep neural network. We just increased the hidden layers. Instead of 2 hidden layers, 5 hidden layers. I wouldn't say actually this is a deep neural network, but okay, we were increasing the detail, and there was a little improvement from NN2 to NN5. This is with 5 hidden layers. You see that there was a little improvement, but we were not still satisfied. Therefore, we moved and analyzed also another number of machine learning methods, such as the support vector machines, the k-nearest neighbors, XGBoost, polynomial regression, and finally the random forest, RF. So RF, random forest, proved to be the most efficient method for identifying and trying to extract a quantitative assessment, so a kind of regression for our LDL equation. And we use as hyperparameters a high number of trees, 1000, and also the depth was rather high, 20, and the splits and the leaf went down up to 1. We used the standard SQRT, the square root, for the number of features, and we didn't implement the bootstrap since we had a high number of different individuals, in excess of 24,000 values for the LDL. And by running these optimized RF, we were able to improve from the neural network 16 down to 10. So respect to the available equation 36, there was an improvement of 71%. So a significant improvement. And in fact, as you can see here at the bottom, almost half of the points, half of the individual are in excess and half in not in excess. And it means that the percentage of points below the equivalence line is almost equal to 50%. So a big improvement, a significant improvement in all the other indicators. And by the way, the percentage of negative points, which would mean negative LDL, that doesn't make sense, is 0 in both situations. But respect to the Friedewald equation, original one, the Friedewald has a big problem. When triglycerides are quite high, it provides a negative assessment in terms of LDL. So going to the conclusions, we were able to show that nutritional data mining is a good activity, trying to extract information from large amounts of data from individuals allowed us to select a candidate nutraceutical that, based on selenium, phosphorus, and magnesium, was able to reduce, hopefully, the gastrointestinal inflammation. And there is also space for personalized nutraceuticals. So once we know the behavior of a large number of individuals, let's say a population, we can also try to target specific nutraceuticals for the features of a single individual, the single person. Future directions: personalized preventive healthcare, patient-specific machine learning models, and a final message is about chemical engineering's unique contribution as from the molecular formulations seen in the first two presentations up to the last two based on machine learning, these and the following one, we can see that there is space for healthcare solutions where chemical engineers are perfectly positioned to bridge the gap between data science and patient-centered nutritional interventions. So I would thank everybody for the participation, and questions are welcome.
Thank you, Davide. Thank you very much for this very, very interesting question. I didn't mention, but I want to do it now, that Davide is a vice chair of our section, and actually from the beginning he was involved in the establishment of this section together with Professor David Bogle from UCL. So you are one of the fathers of these ideas. Okay, thank you. I'm looking if we have any questions from the audience. Yes, we have one. I will read it. It's a very interesting presentation. How were the optimal hyperparameters, that is number of layers and nodes, were chosen in the artificial neural network?
Yes. In both situations, not only for the artificial neural network, but also, for the, for the artificial neural network, but also for the random forest, it was subjected to a number of different tests, and sometimes we were also able to do let us say, a brute force approach, because the identification of a neural network, also the largest one, took, let's say, less than 10 minutes, and for the random forest, less than 300 seconds, let's say 5-6 minutes. So it was also possible to tune the different parameters, but by, first of all, making some tests, reducing the interval of those hyperparameters, and then, let's say, looking at their performance by changing according to a brute force method.
Okay, thank you. I wonder if maybe this question is stupid, or maybe yes, this is another, sorry, I read another comment, a very interesting discussion. Thank you for organization, Lorenzo Teodori. Thank you, Lorenzo. We were happy that you enjoyed this meeting. And I have a question which is maybe stupid, but of course, when you show the data set, big data set, and you show the equation underestimates the level of the LDL, is it possible just to change the equation? I mean, just to change the, probably there are some, numbers and maybe even powers in I don't know the equation, but actually you can adjust the equation.
Yes, you're right, absolutely right. And the equations are very, very, very simple. It's just a matter of a division or multiplication and a subtraction. But the problem is that with the respect of medical doctors, they mathematics is usually not their strength.
Okay.
And also, let us say that these diagrams were almost never shown, also because this data set is rather recent, 2024.
Okay.
And there is also an habit. I mean, probably, let us say that having a new equation might change their mindset. Why? Because they are so used to these kind of biased equations. Just for the sake of, let's say, example, the Friedewald equation that is even worse, significantly worse than the NIH2 is still today used very largely, for instance, in Italy. And
Okay. Yep.
some doctors, also expert of those topics, do not even know the NIH2 equation.
Okay. Okay. So now the way is you have to think somehow how to implement this model. I mean, not a model, is actually the way of estimating the LDL to the doctors. How can they use them?
Yes.
A computer program, or I don't know, iPhone application, just to use it very, very easily. Okay, I mean, I don't want to start a long discussion, but it's very interesting topic, and thank you very much for this presentation. And we have to move on to the last one, which is also about machine learning. And Daniele Pessina, who is from Imperial College London, he will present the talk. So please, Daniel.
Okay, thank you. Let me share.
Yes, we see.
Okay, excellent. Okay, great, thank you. Yeah, today I'll continue the talks about machine learning and modeling has to do with pharmaceutical applications. And I'd also like to acknowledge the other co-authors and collaborators of this work. So Dr. Abbiati, Professor Manca, and my supervisor, Dr. Papathanassiou. So I was at the end. So my work that I will present today is focusing on modeling pharmaceutical transport through the body. So during the development of a pharmaceutical, the practitioners, the clinicians are interested in knowing and examining and assessing how a pharmaceutical travels through the body. And this is known as the ADME process. So you start with absorption, have distribution, metabolism of the drug, and then finally excretion from the body. And then in clinical studies, especially, clinical studies involve human patients rather than animal patients, and they're time-consuming and expensive, and also must consider patient safety with regards to the experimentation. So these clinical studies are used to assess the ADME kinetics of a newly proposed drug, for example. However, the fact that you have these real patients, let's call them, introduces these concerns about safety and how much you can experiment with them. So therefore, it becomes beneficial to have a virtual patient or an in silico model that you can use to examine and query this ADME process for the drug. And this will then enable rapid and parallel virtual experimentation. So it has already been used for model-informed drug development. However, basically the way that we do this, and actually, Tomasz, I'd like to thank you because you introduced this concept at the start of the webinar, but we use these models called physiologically based pharmacokinetic models, or PBPK models. And these models essentially work by splitting the human body in different compartments. So you can see some compartments here to the left. This is actually the model I will present today. For example, here the human body is split into 5 different compartments with the plasma, liver, circulatory system, and different types of tissues and organs. So this is a simplification of the ADME process through the use of ordinary differential equations and parameters, and simply we apply our chemical engineering background and knowledge and kind of methodology to a model of the human body. And with these models, you can then get, ideally, you have patient-specific kinetic parameters, so parameters that describe kinetic transport of the drug between 2 different compartments, for example. And then you can also get simulations simulations and predictions of the concentration of the drug through the human body through time. So this can then be used for example, optimal drug dosage studies. The problem here, however, is that you can identify kinetic parameters for the specific patient only if you have accurate and frequent drug concentration measurements. In practice, however, this often isn't the case. So as I said, these clinical studies involve a cohort of human patients, so quite a wide range of human patients, each with their own characteristics. So each of them has their own height, weight, age, for example. You also face challenges to do with the identifiability of these parameters. So in a maybe typical process model, you might be able to measure every variable through the use of probes in a reactor, for example. However, when talking about clinical studies with patients, you often cannot actually experimentally measure everything. So most of the time, or often you can only measure the plasma, so the concentration of the drug in the blood. This is because some other measurements that might be in the organs or in the liver are actually too invasive for the patient, and definitely too invasive to do on everybody and often. Therefore, we see that the number of per patient measurements is also limited. So if you try to make a model of a single patient, so an exact kind of digital twin, as it's known, of a patient, you cannot formulate it without having high parameter uncertainty. Therefore, the measurements from different patients are all aggregated and used to estimate cohort-wide kinetic parameters. So a single set of kinetic parameters for the whole cohort that was measured in the clinical study. The problem here, however, is that actually this cohort might not be exactly statistically uniform. For example, there might be underrepresented populations or special populations that firstly don't have enough patients to make their kinetics significant in the whole aggregate, but also that in general they present distinct ADME characteristics. And therefore, if you develop a single dosage methodology, these patients will receive suboptimal dosage and suboptimal experimental design of clinical studies. So an example of an underrepresented cohort might be younger patients in a mixed age cohort, or patients with diseases in a mixed health cohort. So therefore, kind of bringing back to what Davide was talking about, we want a precision medicine approach. So we want individualized models and individual ADME kinetics that are that we can then predict. And therefore, the question then becomes, how can we actually assess the model sensitivity to the patient variability? So how can we know which patient features and which kinetic parameters actually affect the model outputs? So here I'm going to introduce global sensitivity analysis, which is a method that is used quite widely in chemical engineering to find the degree to which model outputs are sensitive to changes in the input parameters. So as I said, patients have unique ADME characteristics, and we assume that they have their own corresponding set of kinetic parameters, so individual kinetic parameters. So as you can see here from the figure, you might get different concentration profiles based on the different patients. Therefore, GSA can actually help us answer questions such as which disease features or characteristically significantly affect the therapeutic outcome, or which patient features also affects the model outcome. And also, it can also tell us if the model structure can be simplified. There is, however, a challenge with global sensitivity analysis, especially with PBPK models. So the GSA of PBPK models requires a lot of simulations to converge, so to have reliable results. So you need, in the results I will show here, you need more than 1 million simulations. To give you an idea, to give some context here, generally, a process model needs in the order of 4,000, maybe 10,000 simulations at most. It's very, very difficult that a model will need more simulations to get GSA to converge. And actually, PBK models need 1 million simulations because they're very nonlinear, often have very stiff kinetics, because the model also assumes the model assumes the injection and the dosage of the pharmaceutical, for example. So there is a very, very stiff and discontinuous increase in the concentration when the drug is injected. There have been several methods proposed to tackle these challenges with the GSA. However, they each have their own drawbacks. So some methods proposed include parameter grouping. So trying to group the different parameters and run smaller GSAs in a reduced set. However, this does not consider the whole parameter domain. So you're going to miss out some interactions between parameters as well. That might be useful to know. And there are also other methods known as meta-modeling methods where a kind of non-machine learning surrogate is fitted to the data to perform GSA and decomposed to perform GSA. However, these meta-modeling methods actually scale quite poorly with increasing dimensions. And the model that I will show today only has 8 input dimensions, so the 8 kinetic parameters. And I should also stress that it is kind of like a toy problem. It is amongst the simplest PBPK models you can get. And you might actually have sets of 20, 30, or 40 kinetic parameters that need to be assessed. So here we present our machine learning enhanced global sensitivity analysis methodology. In the reference method, that is kind of what is the standard method to perform GSA. You collect or design a number of GSA simulations, collect a matrix of n GSA parameter combinations. So in this case, a matrix of 8 columns and 1 million or more than 1 million rows, and essentially collect this matrix and perform GSA after you have simulated these all of these samples. So each combination of kinetic parameters. In our methodology, instead, we drastically reduce the amount of simulations that need to come from the PBPK model itself. So we simulate a much reduced set of simulations that are actually then used to train a machine learning model for each of the relevant model outputs. And then after that, this machine learning model is used and replaces the PBPK model, and is used to simulate the combinations needed for GSA, the combinations of parameters needed for GSA, and return GSA results, so sensitivity analysis results. To give more information on the mechanistic model that I'm going to show today, the model is split into, again, as I said, these 5 compartments. You have 8 kinetic parameters, which are these labeled ones here in the table, and these all consider transport of the drug and the ADME kinetics themselves of the drug through the human body. This model also includes a single bolus intravenous injection at the start of the model simulation. So already a simplification compared to other models that might have multiple intravenous injections. And there are 5 scalar model outputs. So we consider here the area under the curve output, so the integral of the concentration profile for each output. So for the concentration in each compartment. However, this methodology also does work with time-indexed output, so time series. In terms of the machine learning models, these are the same that Davide talked about earlier. So neural networks, we use neural networks and random forest ensembles. And critically, we train them here with 4,096 training points. So a lot fewer, and I will talk about this later as well, but a lot fewer than the 1 million needed to perform GSA with a mechanistic model. And I should also specify that, firstly, we did try other machine learning models, such as Gaussian process regressors and radial basis functions. These didn't actually work very well. And then the hyperparameters of both of the structures were identified via hyperparameter tuning and cross-validation. So we simulated an extra test set from the model to do hyperparameter tuning. So starting off with the reference GSA methodology, I'll present here now the baseline results. And these are basically what we want to replicate with our ML model. So we want to reach the same conclusion that we reach with the mechanistic model. GSA works based on, or returns these Sobel sensitivity indices. So these three here. Here I'm only going to talk about the total order indices. So the total order indices essentially explain or quantify the degree to which one changing parameter affects the model output, and disregards any other parameters. However, GSA actually gives you more sensitivity indices, and all of them are very useful to examine the model, assess the model, and probe the model structure itself. And then it's important also to highlight that the GSA method needs to converge for the results to be confident and to be actually considered true. So we call a converged GSA, we call it converged when the sum of first and second order indices for all the parameters approaches 1. So if it's lower than, generally the cutoff is 0.97, for example. If it's lower than that, you can probably consider your GSA to not have converged, and you need more simulations to get it to converge. So an example here is in the top table, you can see the GSA convergence sums for each output with a mechanistic model. And with 300,000 simulations, some of the outputs still have not converged the GSA. Therefore, we need more simulations, and we needed 1 million and almost 200,000 simulations from the model to have a converged GSA. And then here we find 3 critical parameters for 4 of the model outputs and 5 critical parameters for the last model output. And by critical parameters, I mean parameters which affect, whereby a 2% change in the inputs affects more than 5% of the model output. You can set the cutoff based on your own model or based on your own engineering judgment, but in general, this is an indication of which parameters are experimentally identifiable with the measurements during a parameter estimation procedure and which ones are not. In the table below here, instead, you can see the total order sensitivity indices for each parameter and for all of the outputs. And in bold, I put the critical parameters. So back to our methodology, to the machine learning enhanced GSA. These first results include the neural network derived GSA results. So this table here shows the difference between the total order indices given by the mechanistic model and the neural network model. So ideally, you want a converged GSA and 0 or practically 0 differences in the predicted indices. So with the neural network, we find that actually the same critical sets of parameters are identified much quicker than the mechanistic model. And the sensitivity index errors are limited to between 1 to 3% at most, and we can consider them to be identical to the mechanistic derived results. So we reach the same conclusion of parameter significance and criticality, and would, the same conclusion with respect to the GSA of the mechanistic model. To give you an idea of the runtime improvements with our method compared to using the mechanistic model, as I said, you need 1 million simulations for the mechanistic model, or more than 1 million simulations. So that took 14,000 seconds, which is approximately 4 hours of computation. So that is for one single GSA run. The neural network enhanced methodology instead took 300 seconds, so 5 minutes of computation time. And this also includes the training of the neural net. Whilst in practice, you can train it once and then just save the trained neural network and launch the saved one. So the training also is approximately half of the time of the runtime. So this would again split the runtime in half. Instead, if we look at the random forest results, the random forest has needed a few more samples to fully converge the GSA. So we had to go from 1 million to 200,000 samples to 4 million, to over 4 million samples with the random forest machine learning model. However, you still have a very considerable computational speed up that you can achieve. This is also because using the random forest to predict a single output, or in any case to perform a simulation, is much, much quicker than using the mechanistic model, because these are, again, as I said, very stiff and nonlinear models that are relatively or quite challenging to solve efficiently. We did see a greater mispredictions in the sensitivity indices compared to the mechanistic model. And actually, we missed a single critical parameter for one of the outputs with this methodology. However, I think it's important to also note that that parameter was on the boundary of being called non-critical. So any kind of misprediction would have made it non-critical, and I think any amount of engineering judgment would have just told you it's close enough to being critical. And in terms of the runtime, again, the random forest, including the training, which is quite short anyways, including the training took 965 seconds. So that's around 16 minutes of computation. So again, the neural network was 5 minutes, random forest was 16, and the mechanistic model, so the standard method, took 4 hours. So a massive increase in computational speed up. So to conclude, GSA is an extremely valuable exploratory tool. So as I said earlier, GSA can be used to examine the parameter identifiability of the model. So you can structure and optimally structure your experimental design and the clinical studies. And this is very important because, as I said, these clinical studies are very, very expensive, both from a time point of view, monetary point of view, and in general, the cost of the patients, because you don't want to subject them to unneeded testing and measurements. It's also useful to then explore which parameter features affect the therapeutic outcome. So this could be if a patient has a renal failure, for example, you can perform a GSA around those relevant parameters, kinetic parameters, and examine if that will actually have an effect on the therapeutic outcome. So therefore, we propose a machine learning enhanced workflow for GSA that can reliably capture parameter interactions with a much limited set of training data, much fewer samples compared to those needed from the mechanistic model. And we can reduce the GSA execution time by over 95% compared to the standard method. And our future work is on rapidly generating cohort-specific GSA assessments to support clinical trial development, and in general, introducing more personalized aspects to the modeling of the ADME process and to PBPK models in general. So with that, I'd like to thank you for listening, and I'll be happy to answer any questions.
Thank you very much, Daniel. Thank you. Very, very interesting talk, showing another aspect of system engineering, let's say, to biology or to human body, to pharmacokinetics. Okay, I will see if we have any questions from the audience. I cannot see anything. I have a question, because your data, your output data, were generated by mechanistic model. Then you try to find if this machine learning can predict accordingly and that it's faster. But in reality, how can you get this amount of data? I mean, from the clinical studies, this is what we should look for, or this is not needed here?
So the GSA focuses specifically on the model itself. So the results give you an indication both of what the model is kind of telling you in terms of maybe some parameters are more influential than others. So you see age is more important than height, for example. But it also tells you about the parameter identifiability itself within the model. So you assume a model structure, and the question becomes essentially, given this model structure that I'm proposing to be accurate and that I'm proposing will accurately predict the drug concentration in the body, given the structure, can I actually estimate these parameters, or will I get just a lot of uncertainty? Okay. And then linked to that, you can then also fix parameters. So if you have, as I said, more complex models have maybe 30 kinetic parameters, you might find that some are actually very non-critical and don't really affect the outcome in any tangible way, and therefore you can fix them. And by fixing them, you reduce the amount of dimensions that you have in the model, and you can therefore then improve your parameter estimation of the model.
Okay, thank you very much. Thank you. I will see again no questions, so I will summarize our meeting. Thank you, Daniela, and thank you all previous speakers. It was really kind of broad perspective. We started from let's say drug delivery systems or drug carriers. Then we went through the elimination of toxins from the body, and then we finished into different but interesting modeling applications to medicine, to different aspects of medicine. So it shows that chemical engineering is a very broad discipline, and we can use our capabilities to solve these physiological problems, and it is very, very encouraging. And I hope that all the listeners who are online also see this very big perspective for chemical engineering. I want to attract these people to our community. Last but not least, I want to thank the organizers, the European Federation of Chemical Engineering, and especially to Martin Poe, who was responsible for organizing this, all the spotlight talks, but also our meeting. And I hope that we can meet in person very soon, maybe in Lisbon during the European Congress of Chemical Engineering, and maybe in other way. But I want to also thank Boelo, Schuur, who opened this session, for being with us, and I hope that you see that this section is active, and we want to do our best to push chemical engineering to new directions. So thank you very much.
Thanks a lot, Tomasz, and to all the speakers. It was a wonderful kickoff of this 2025 spring season of Spotlight Talks. And with this, I would like to wish everybody a nice evening, and hopefully see you in the upcoming Spotlight Talks in the coming week, or indeed in Lisbon. Thank you. Good night.
Thank you.
Thank you.
Thank you. Bye.